Let us discuss T lymphocytes. So as we suggested, T lymphocytes become immunocompetent in the thymus. The idea of immunocompetence is that they are able to recognize a non-self antigen binding to another self cell. So they can find pathogenic antigens, but only when they are binding to self. When we have the idea of the development of these guys, we're talking about no, only about 2% of the T lymphocytes will survive the process of immunocompetence inside of the thymus. They are only the cells that have the sharp ability to identify foreign antigens at the same time as recognizing self without binding too tightly. So just to put like a fine point on this, T lymphocytes will not bind to anything until they recognize a pathogenic antigen and a protein from within the body cell, which are usually bound together. So when we're talking about this, we have to go back to the idea of the antigen-presenting cells. Get back to this figure here, right here. The antigen-presenting cells are cells. We've called them phagocytes up until this point, but now you have to kind of turn around and remember that these guys have some knowledge. You know, not only have they consumed these pathogens, but now they own some of the pathogen. Like the pathogenic antigen that they consumed, they are now able to use that. So they engulf these antigens. They're able to present the fragments on their own surfaces rather than just releasing them. They may actually take these little green bits and present them on the surface. This in turn will help the T lymphocytes will be able to recognize this. So remember, self and non-self at the same time. That is very important to T lymphocytes. This part here. Okay, so now we have to discuss the idea of the major histocompatibility complex, the MHC, major histocompatibility complex. So there are class 1 MHCs, which are proteins that are found on every cell of the body, and then there are class 2 MHC proteins, which are only found on the surface of antigen-presenting cells themselves. And we'll take a closer look at these in class. I think it's better just to talk about it in person rather than trying to discuss the whole process. However, I would encourage you, of course, to go into your book and read up on the MHCs to try to at least get an intro to this stuff. It is a rather complex concept, so we will be talking more about it together. So in this class 2 MHCs doing their work, class 1 MHCs doing their work. And yeah, I think we'll just talk about it together. We'll kind of draw out the whole process here. But the basic idea is that this is presenting material pertaining to pathogenic antigens. And only when it is presented can the T cells be activated. Okay, T cells will activate by 2 methods. First of all, you have to have, number 1, you have to have antigen binding, which remember means that you have both self and non-self together. And 2, you have to have co-stimulation, which allows for the completion of T cell activation, or in some cases, abortion of the process if there is no co-stimulation involved. Usually the cofactors are going to be other cell surface proteins other than the MHC. The T lymphocyte will enlarge after it has had the antigen binding, after it has had the co-stimulation. It will enlarge, it will form clones. Here are the clones. The cloning of the cells will peak at about day 7 and will die off at around day 30. So it takes a little while for the T lymphocytes to become, large in numbers so that they're actually affecting change within the system. But it is a very, very powerful system, so you don't want to activate it arbitrarily at any given time. Like I said, we'll just go through this in class. Take a look at it in the book. Take a look at this picture. Maybe you want to try to draw this and see if you can kind of suss out what's happening before we actually get to class, because better to have knowledge previous, even if it leaves you with lots of questions. It is better to have that knowledge, first of all. Okay, so T cells are able to display two different types of glycoproteins on their surface, either CD4 proteins or CD8 proteins. Cells that display CD4 are called helper T cells. Those that display CD8 glycoproteins are called cytotoxic T cells. And then just so you know, there are also suppressor T cells, and there are also memory T cells and others which are less numerous. And of course, just refer to this figure. You want to fill it in maybe in your blank figures. Maybe you just want to draw it by hand in your book. It would force you to learn a lot of the information, but we will be drawing a lot of this stuff together as well. Okay, so let's talk about the helper T cells. The helper T cells are what we call the managers of the immune response. They have three roles. They bind to B lymphocytes that are already displaying the antigen fragments. We'll be talking about the B lymphocytes in the next video. And they help the B lymphocytes to replicate. What we're going to be talking about in the next video is the idea that B lymphocytes replicate on their own. But in fact, if helper T cells are in the area, the B lymphocytes actually replicate much more quickly and in much greater number. So, secondly, the helper T cells are able to release interleukins and other cytokines. There's the cytokine word there again, which remember, cytokines do all kinds of stuff. They enhance the enhance the inflammatory response. They help to enhance white blood cell levels. They help to attract white blood cells into a region. And thirdly, helper T cells help to enact cytotoxic T cells. So cytotoxic T cells, next, are killer T cells. They're sometimes called cytotoxic T cells and sometimes called killer T cells, sometimes called T sub C. And these can directly attack and kill other cells. So, for example, if you have infected cells of your body that have taken on a virus or a bacterium, or a cell that is cancerous, cytotoxic T cells are actually capable of killing them off, which is extraordinary, isn't it? So this is how they work. The T cell will bind tightly to its target cell. Here we have a cytotoxic T cell. Here we have a much larger cancer cell here, you see? But there's a few cytotoxic T cells in the region. So they all start going to work on this cancer cell. They start to, they bind tightly to the target cell. They release perforin chemicals, these perforins here. These form transmembrane pores, and here they're referred to as perforin rings. And then they help to, they release these structures that are called granzymes and lymphotoxins that enter the target cell through the pores that have been created, and then they degrade the cellular contents, and this stimulates apoptosis. So again, I think I just want to reiterate. Helper T cells have three roles. Well, they're the managers of the immune response. That's kind of what they are. They have three roles. They bind to B lymphocytes and enhance their activity. They release cytokines, and so that enhances a whole bunch of stuff. And thirdly, they help to mobilize the cytotoxic T cells. And then the cytotoxic T cells themselves, if you're just looking at their role on their own, they bind tightly to target cells. They release porphyrin chemicals that perforate the pores of the structures they're attacking. And then they release these chemicals called granzymes and lymphotoxins. And that's the T cell story.